Hey guys, in this video we are going to talk about the mechanisms for all of the antimicrobial drugs. We'll start with the antibacterials and move on to the other microbials in the next section. There are a lot. I'm just going to start off this video saying there are a lot of drug names in this video. I did not want you to know all of the drug names in this video. This is not a pharmacology class. I do want you to know the modes of action, all the different ways that they can impact the bacteria and how they end up being bactericidal. So that I do want you to know. If I have specific drugs that I need you to know, they will be in the test review, okay? In the exam review, look for those. I'm going to mention lots of them in this section, but I do not want you to stress about knowing every single drug and about how every single drug works. That is not what I want to focus on in this class. So we're going to briefly go through the different ones and how they work. We mentioned dosage and method of transmission, how the person is going to obtain this medicine, and now we'll talk about all the different ways that they can attack specifically the bacteria. So with the drug, you want it to be selective when it's toxic. We want it to only be toxic to the bacteria, right? We don't want it to hurt us. And so it's going to be a fine balance of trying to have enough medicine to kill off the bacteria without hurting the human being that is exposed to this drug. And we'll talk about the side effects on some of these drugs. Some of these drugs are no longer used because they have some pretty harsh side effects, but we'll go through those. But we do need to know there's selective toxicity. Are they only going after gram positives or gram negatives? Is it a broad antibiotic that has major sweeping, is going to kill off everything kind of antibiotic? What is it, or how selective is it? And then again, what is its mode of action? So is it attacking the cell wall? Those are the ones that we're going to talk about first, the ones that attack the cell wall, or they could go after the membrane or the ribosomes, because we talk about central dogma, right? Central dogma is DNA, and then to RNA, and then to proteins. And if you mess up with the ribosomes, you can't translate that messenger RNA into the proteins that it needs, and the bacteria will die. So there's lots of different things. Ribosomes, metabolic pathways. If you can't mess with the DNA or the RNA, you're going to stop it at step one of central dogma. So there's lots of different ways for us to kill off bacteria, and then want you to know the different modes of action. So we'll break it down. The first one is the cell walls, going after cell walls. And so we have a table here of all of the different ones broken down by modes of action: cell walls, proteins, membranes, nucleic acids, going after the antimetabolites. Some of these things will go after like folic acid or something like that. Some of them go after, if we're talking about TB, they could go after the mycolic acid. They could be going after metabolites. So there's lots of different modes of action. That table summarizes them all kind of in a nutshell in broad terms. If we're going about the wall, the cell wall, on these bacteria, the first group of drugs that attack the cell wall is one of the very first ones that we discovered, that we learned about, penicillin. Remember we talked about how he went on vacay and came back, and the penicillin mold got onto his plates of staph and had a zone of inhibition. And so that was one of our very first things. This one attacks our bacteria with these beta-lactam rings. So everything in this group has this ring. It's in blue, this ring right here. That's its beta-lactam ring. And what it does is it's going to mess with the structure of the peptidoglycan in the cell walls. So we said every bacteria has peptidoglycan. Gram positive had more peptidoglycan than the gram negative. So the gram positives, since beta-lactams are going to mess with the peptidoglycan, are more affected from these beta-lactams, but some of these will still get some gram negatives too. I want to say penicillin G, the textbook says penicillin G and penicillin V, also get some gram negatives like Pasteurella multicida. But beta-lactams, they all have this ring. We've got penicillin as our beginner, but then remember we talked about how once we figured out the structure of penicillin, that we could make and modify all sorts of derivatives. So we've got Keflex over here, Monobactrim, and then carbapenem. So in the last section, we talked about what's her name? It was Roberta. And that she had an endoscope and it was covered in bacteria, and that bacteria was carbapenem resistant. So she was taking this drug, but this drug she was resistant to it, that bacteria was resistant to this antibiotic here. But all of these are beta-lactams. Beta-lactams attack the peptidoglycan. So our gram-positives with all that peptidoglycan, beta-lactams will interfere with those. So penicillin, Keflex. There's a new version of cephalosporin that they can use to attack MRSA. We're going to go into MRSA and the drug resistance in the next video. We'll get to those. Some of these are coming from other areas. Vancomycins are coming from actinomycetes. Bacitracin is coming from bacillus, so it kind of makes sense why it's named what it's named. So some of these are coming from other bacteria putting out these chemicals to kill off everything around them. So all of these are beta-lactams. Where the beta-lactams attack? Cell walls. They attack the cell walls. So there's a whole bunch in this table over here, lots of them, and we can see if they're a narrow spectrum or a more broader spectrum. But gram-positives in general, beta-lactams will go after them the most because it goes after the cell wall filled with peptidoglycan for our gram-positives. There are lots of them in here. Again, I'm not going to expect you to know every single one of these. Listen to the test review on specifics. What about if we mess with the proteins? Protein biosynthesis. Protein is going to be made from the messenger RNA. Got to meet up with the ribosome. The ribosome has to have both pieces, the little piece and the big piece of the hamburger bun that I called it. And then when they meet up, we're going to have the transfer RNA inside of there. That's going to bring every single amino acid and it's going to create the protein. So we can interfere with protein making in the bacteria, and then that's going to shut down their central dogma, and that will kill the bacteria too. So we have aminoglycines and tetracyclines. Both of these are going to bind to the small part of the ribosome because we have the small part that's on the bottom that's going to meet up with the messenger RNA, and we have the big part at the top. That's the 50S and the 30S. 50S is the big part of the hamburger bun. 30S is the small part of the hamburger bun. Chloramphenicol, macroslides, linkoslides. Some of these things, like the aminoglycosides, are going to have side effects. Like we were saying, you need to balance out, is it worth it to give this person this medication knowing that they have these side effects, and does this person have pre-existing conditions? So we wouldn't want to give them this medication at all. So aminoglycosides have toxicity to the kidneys. They are nephrotoxic. They could be neurotoxic and damage the nervous system. They could be ototoxic and damage the ears. So it's things to think about. Tetracyclines were something that used to be given out quite a lot, really common. And I have colleagues my age and older that were given tetracyclines. And when they were developing their teeth, because they were a little bit older and they were given this antibiotic, I don't know if you've noticed some people have this, but their teeth were discolored as they were developing and they were given this antibiotic. Their teeth are not white. They're discolored, kind of a yellowish, grayish color because of tetracycline. That's one of the side effects. Phototoxicity is another side effect of tetracycline. So some of these are wonderful and great, but the fewer side effects, the better. Azithromycin is going to attack that 50. We have the list of it in here too. There it is. Azithromycin. So that's going to attack the big part. This guy on the ribosome. Azithromycin also has something that when we were talking about how long does this drug stay in the system? Is this a drug that stays in the system for like an hour and a half? Or is this a drug that stays in the system for much, much longer? So this one's can stay, a Z-Pack stays in the body for a very long time. So it allows you to just have the 1 dose a day and a 3-dose therapy course, and then you're done. So it depends on the drug and how long does it stay in the system. But Azithromycin will attack the big parts of the ribosome and lead to death of the antibiotic. Chloramphenicol is in here. There it is. It is another broad spectrum. Chloramphenicol had some terrible side effects. I was reading in the textbook, and it said that it was used to treat everything with a wide range of infections. If you've got a bacterial infection, we give you chloramphenicol. Everything from, what did it say, meningitis to typhoid fever to conjunctivitis. So top to bottom. But the side effects, if they were given to pregnant women, it led to birth defects. It led to lethal gray baby syndrome, suppression of your bone marrow. So some seriously terrible side effects from this antibiotic. So it's things that we need to think about with all of these drugs. What are the side effects of these? Membranes. We've got polymyxin, and this one is usually on the polymyxins. We're going to mix these up and topically prepare to prevent wounds. And so it's a narrow spectrum. It's going to be gram-negative bacteria, and it's going to kill the membrane. So it's kind of disruptive, like a detergent, where we talked about how dish soap will break up fats. That's kind of what this acts like. So it's going to put holes, it's going to put pores in the cell membranes, the polymyxins do, and that's going to kill off the bacteria. And daptomycin does a similar kind of effect where it attacks the cell membrane of the bacteria cells. Nucleic acid synthesis. Where we are. Got rifampin. That one is going to, this one starts with an R. Rifampin is going to start with an R, and it's going to inhibit RNA pol. So it can't lay down new RNA if it can't produce the RNA polymerase. So rifampin will stop that, and it says that we can use that in a combo, in a drug combo. So if we were treating, here we go, TB, because TB is getting more and more drug resistant, so we're having to use drug combinations. Rifampin is one of those that we can use in combination treatment, and rifampin starts with an R. It is going to attack the RNA pol within that bacteria. And then the fluoroquinons, our Cipro, is going to attack DNA gyrase. So DNA gyrase is a type of topoisomerase that we talked about that's going to kind of unwind DNA before it undergoes replication, and then its topoisomerase shows up again at the end. And so if we block that with our fluoroquinons, then we can't undergo replication. So both of these, rifampin and the fluoroquinolones, are going to attack nucleic acids. Rifampin's RNA and the fluoroquinolones are DNA. What if they attack the metabolic pathways? So some of these things, the sulfa drugs that we mentioned before, are going to inhibit folic acid synthesis, so they can't create their own folic acid. That's something that we need to bring in anyway, so that attacks the bacteria and it doesn't affect us with the sulfa drugs. So we mentioned sulfa drugs before. They are some of the oldest that we talked about coming, and they are going to be sulfonamides. They are going to be anti-metabolites, so they are going to block, here we go, I'm going to block the PAVA. It's going to stop the synthesis of our folic acid. So depending on what it is, it will block it. So sulfonamide is going to block it right here, and then this guy is blocking it here, and eventually this guy is going to block it over there. Antimetabolites will block different things. Some of them are going to block folic acid. Some of them may be blocking something else. We can use these for different treatments. Yeah, this one is talking about how we can use this one for tuberculosis as well, because it is mycolic acid synthesis is eventually what it's going to produce at the end. And we already talked about the sulfa drugs are going to help with folic acid synthesis. So lots of different ones mentioned in here. I'm not going to go into specifics on these metabolites. So let's check in with Marissa. The doctor's going back through her medical history. She saw that they saw that their hospitalization in Vietnam, because remember she had her spinal compression surgery there. So while she was under for her spinal compression surgery, she was catheterized and she got some antimicrobial drugs. And so we're going to do a CT scan of Marissa's abdomen, make sure that she does not have appendicitis since she's in that abdominal pain, and see what's going on in her blood work. She's got elevated white blood cells. We're going to look and see if she's got urinalysis and urine culture to see if she's got a UTI. We're looking for white blood cells, red blood cells, bacteria, what's going in, what's growing in her urine. They cultured it. It comes back positive for bacteria. So she's got a UTI. So she's going to get Cipro, and they're going to treat her with that antibiotic, culture her future urine, and see what is going on, see if that took care of her UTI. So let's talk about the other guys. First, we talked about the bacteria. Let's talk about all the other guys. Maybe our helminths, our fungi. So the first ones in this section, they were talking about fungi. And so if you've got a mycosis, you've got a fungal infection. All of our eukaryotics, it was tough because fungi is a eukaryote. So it's hard for any of these eukaryotes, fungi, helminths. How do we attack that thing and not kill the parts that are all found in all of our other eukaryotic cells? How do we be that specific to structures just found in fungi? And one of the ways that we can do that is to go after the ergosterol. So we said that our membrane for our cell membrane is a phospholipid bilayer. And so we've got all those little phospholipids in there. We've got proteins in there. We've got carbs dangling in there. But we also have another lipid in that membrane, and it's cholesterol. Cholesterol for us in our eukaryotic cells is going to give us structure. And we're going to use it in our membrane. Fungi have ergosterol, so they have a different molecule. So if we can go after that molecule, it's not going to damage any of our human eukaryotic cells. It's just going to go after the fungi's eukaryotic cells. And if we damage that ergosterol, then their membrane's going to break down and they'll die. So there are different types of antifungal drugs that can do that. So we have imidazole, lemon. If they interrupt those, then we're going to break them. We're going to break up the membrane, and it will stop. So fluconazole, there we go, got it, is one that will help with ergosterol biosynthesis and stop it. Imidazole, allylaminines. And then there is another one with our echinocandins, and they are going to prevent the synthesis of our beta glucans right here. So sometimes we'll, just like we were talking about with the bacteria, are we going to talk about breaking down the cell membrane? Are we inhibiting DNA and RNA? We're going after the nucleic acids. Are we going after their mitochondria? So there are drugs that can attack the quinones. When we talked about cellular respiration, we talked about there was glycolysis, and there was citric acid cycle, and there was electron transport chain, and the electron transport chain played hot potato with the electrons. You take it, you take it, you take it, you take it. Well, with this drug, not the quinone, we can mess with the quinone's proteins that are found in the membranes of the mitochondria in the fungi. So super specific, and that's what we need. We need to be very narrow spectrum, and that's what that one goes after. So we can go after membranes, we can go after nucleic acids, we can go after metabolites, similar plans of attack with fungi. How can we go after these structures? So lots of them for the ergosterol synthesis. That's going to include medicine for thrush, yeast infections, cryptococcus meningitis, fungal skin infections. So all of that, ringworm, jock itch, that can be treated with drugs that go after the cell membrane of the fungi. Then we have nystatin, that's going to create pores in the cell membrane. Capsifungin is cell wall, microtubules. All right, moving on, out of fungi. And we have a mini test case. And this person's name is Jack. He is 48. He has HIV. He has been doing antiretroviral therapy to keep his HIV under control. He had a really stressful week at work. He came home, started to get a fever and a dry cough, and thought it was just a flu, tried to relax, take it easy. But then he started experiencing fatigue, weight loss, shortness of breath. And so he went to the doctor. The doctor ran a pulse ox, and his oxygen levels are low. So then they did blood testing, x-ray to see what's going on with him and his cough. And he had cloudiness on his lungs in his chest x-ray. And they checked his sputum and were able to culture a bacteria. So he's got pneumocystis pneumonia. It's actually not a bacteria. It is a fungi that was previously a protist. So this is a sneaky little thing that kind of has characteristics of all sorts of stuff. But pneumocystic pneumonia, PCP, is caused by this fungi. So they are going to give him some medication. They're going to give him Bactrim, and hopefully that will knock it out because PCP infections with people with HIV results in 10% of HIV-related death is because of this infection from this fungi. So they were able to give him medicine and see how he was doing. And after he was given the medicine, his white blood cell count started to go back up. So Jack is making a recovery. They adjusted his antiretroviral therapy to help with that, and he is making a recovery. So let's move on to the protist, because PCP was thought to be a protist, and now it's in the fungi group. Let's go on to the protozoans. So all the different protist stuff out there. So there's a lot of different medications out there for malaria. Specifically, we've got chloroquine, been making the news lately. I won't go there. But it is one of the drugs that can treat the parasite that comes along and is spread from the mosquitoes that causes malaria. There are side effects to these drugs just as well as there are the side effects to the antibiotic drugs. Some of these things can cause anemia. Some of these can cause heart issues, specifically with the quinolones. They are going to cause possible hallucinations, cardiac issues, and possible anemia. So lots of different ones. Proquinol is also another malarial one. Artemisinin malaria. Metronidazole and tinazidol is going to treat giardia that I was telling you about that you can get from drinking contaminated water, as well as trich, which is the only protist caused STI, and it can also treat Antimicrobial histolica. So lots of different anti-protozoan drugs. Some of them are going to inhibit the mitochondria. Some of them are going to inhibit the metabolics like the folic acid synthesis. Some of them will inhibit DNA, and the chloroquines are going to inhibit the hemoglobin deoxification. So multiple drugs that can treat the protists. What about the helminths, the bigger guys, the things that we can see with our naked eyes? We have roundworms. If you have treated your pets for worms, you have used this medication on your pets. So ivermectin is going to treat against roundworms. We have niclosamide, can help with tapeworms. and schistosomiasis. Schistosomiasis can be treated with praziquantel or a whole bunch of other possible drugs. Praziquantel is going to attack with calcium, and then the other three drugs can attack their RNA synthesis. The nicotinamide attacks ATP, and its production going from ADP to ATP, it's going to inhibit that. It's going to block the ATP synthase pump. The ivermectin that we mentioned is going to starve out the helminths, starve out the worms, and that they will eventually starve and die. And sometimes if it's a dog and it's got heartworms and you treat them with this, it kills off all the worms at the same time, and that can lead to a blockage in the circulatory system that passes on to blocks, maybe it blocks up the lungs or someplace else when you treat a dog that's got a heavy dose of heartworms with ivermectin. So these will treat heartworms. They're going to treat river blindness. Other things that it can also treat mites and lice, as well as bed bugs can be treated with ivermectin. What about viruses? We mentioned that Jack, the patient before with PCP, had HIV. So what are some of the things that we can use against viruses? So some of our antivirals, acyclovir is one, and what it's going to do is it can help with herpes infections, and pretty much it's going to cause, there it is, chain termination when DNA replicates. So remember when we talked about the Sanger method, and every nucleotide ended up ending, it was missing that hydroxyl group at the end, so it had to end every time it went to replicate. That's what this is doing. So acyclovir is causing the cells to not be able to undergo mitosis, not be able to copy themselves for DNA replication, and it stops that. So that's what acyclovir does. It's marketed as Zovirax. If anybody is being treated with that, that's how that works. The antiretrovirals. So Jack had HIV. He's getting this. He's getting the antiretroviral therapy. And so we mentioned that HIV is going to be an RNA genome. And so that RNA has to back up and go back to DNA. So it brings in its reverse transcriptase that helps it go from RNA back to transcribe back to DNA. So the antiretroviral therapy can stop that. We've got a lot of different ways to stop viruses. Sometimes we can stop them from fusion, just stop them from even getting in the door right here, because we mentioned that you have to have that specific receptor, and that some segments of the population didn't have that receptor. There's also where we can stop the viral if we stop its reverse transcriptase. She had a bad dream. If we block the reverse transcriptase, it can't go from RNA back to DNA, so we can stop it that way. And then we can also stop protease inhibitors. We can break down the proteins that it needs to repackage itself and put itself back out there and go make new virions and infect the other cell. So antiretroviral therapy has a couple of different ways to tackle tricky things like viruses, like AIDS trying to get in the door. Another antiviral therapy is something like Theraflu. We're talking about COVID, but the flu was also going around. It might still be going around for the flu. And so that's going to prevent the virus of actually getting out of the cells. So you've got the flu, it's invaded your cells, and now you're positive for a flu, whatever strain you got. My oldest had flu B a couple months back. And so he's got flu B, and as soon as you test, the sooner you test, the sooner you can take Tamiflu, and you can lock it into the few cells that were infected, and it can't go and infect new cells. So it shortens the duration, is how that works, of the flu, if you can make sure that you get the Tamiflu in early enough. but there are side effects to Tamiflu too, so be sure to consult. We have all sorts of medications in this table over here. The acyclovir that we mentioned for herpes. AZT is going to be one of the ones that helps with HIV. There's another one for herpes. Lots of ones for HIV. HIV has lots of antiviral drugs, and they are very, very costly. It's part of the reason why HIV is spreading in certain areas, not because of lack of education, but because of the expense, because of the cost to treat it and keep it under control. So we can see all of these here that are used to treat HIV just in this list right here. We have 1, 2, 3, 4, 5 drugs right there. And you can see all the different ways that we are trying to tackle it so it can't bind on, so it stops integrating in. We break down the proteins. We're going to stop it with non-competitive inhibition. And then over here, we're going to stop the nucleic acid synthesis. So multiple ways, like we have in this picture here, different ways to try and stop HIV in lots of different stages. So we are going to talk about drug resistance in the next video. Again, don't stress about every single drug. There's so many drugs. But I do mention a few very specific ones in the test review. So focus on those, focus on the modes of transmission. How many different ways can we tackle this? Are we looking for cell walls? Are we attacking the membrane? Are we attacking its metabolics? Are we attacking its proteins? How can we attack this substance, this microbial? How do we go out and try and make sure that it's not going to continue to spread and we can kill it off?